to introduce our very own Chris Solomon. Chris is a limnologist who has been at Cary for almost 10 years now. His work focuses on lakes and other aquatic systems where he addresses large-scale processes including ecosystem function, carbon cycles, and the effects of terrestrial input on food webs. So Chris's training and academic career includes some of the most renowned limnology centers, from undergraduate training at Cornell University to a PhD and postdoc at the University of Wisconsin-Madison to an academic position at McGill University. So fun story, I first came across Chris's name and image in a really unorthodox place. So I learned about him from my husband's photo album back when I was a sprightly undergraduate. Because in a bit of odd history, Chris and my husband both studied and did field work together when they were at Cornell. So then I started graduate school, and when I entered the field of aquatic ecology, I learned more about Chris through reading his papers, and became really inspired by both his research and his productivity. So today I'm just absolutely delighted to have him as a colleague as well as a family friend, and I think everybody here at Cary feels very similarly. So I'm pleased to introduce Chris. The title of his talk today is The Metabolic Regimes of Lake Ecosystems, and I have an important note, which is that there will be no happy hour today, but there will be some food provided in the lunchroom. So if you want to come and celebrate our speaker, drop on by. There will be pizza and salad. All right, take it away, Chris.
Thanks, Tara. Tara was kind enough not to say too much about what that picture looked like with Lauren, so we'll leave that there. And I'm going to start with an apology, too, not for my voice, but for the fact that I'm here. Danielle Hare was originally scheduled to talk here. As you probably know, she just started a new job this week and couldn't quite swing given this talk, so you're stuck with me instead. I apologize. I'm going to give you a talk that I was going to give as a lunch bunch talk last week or this week or something. I was scheduled to do one of those. It may be that it ends up a little bit short. We'll see. So the bad news is it's not a lunch bunch talk, and so you don't have your lunch to distract you while you have to listen to me. The good news is maybe we're short and we get to lunch a little faster anyway. We'll see how it goes. Okay, so I'm going to talk today about the metabolic regimes of lake ecosystems. I'm going to start by thanking a bunch of folks. This is a talk that's basically a visual version of a proposal that we just put in recently and work that we're hoping to do over the next few years. The proposal was written together with Stuart Jones, Bella Alexa, Kathy Weathers, and Paul Hansen. Two of these people are in the room today. Bella was a postdoc here several years ago. Many of you probably know Bella. And then Tara Stewart Merrill, who gave a bunch of feedback on the proposal, and we're hoping to rope into the work should we get funded. And beyond that, I'm going to start with some definitions. Excuse me. We're going to talk a bunch today about ecosystem metabolism, and so just to kind of get us all on the same page, here's an ecosystem. We've got a tree, we've got some roots, we've got soil microbes, Jane, shout out for soil microbes. We've got a giraffe who looks like they might eat the tree there. And the interesting things here for our purposes are we have some gross primary production happening, right? So the sun puts energy into this ecosystem. The primary producers capture that energy and fix it in the form of chemical bonds. And then we also have a thing happening that we call ecosystem respiration. So that's the fact that everybody, the primary producers themselves, both in their photosynthesizing tissues and via their roots, soil microbes, giraffes walking around, all these things are using the cellular process of respiration to release the energy stored in those bonds. And so that's kind of the other half of the equation of ecosystem energetics. So we're going to be talking a lot today about gross primary production and ecosystem respiration. Those matter in sort of that context of like, okay, this is how ecosystems process energy. They also matter because they're a big part of the global carbon cycle, right? Here's a figure from the most recent big IPCC report. And these big arrows here talking about carbon fluxes between terrestrial ecosystems in the atmosphere or ocean ecosystems in the atmosphere. A big part of those arrows is driven by the balances of gross primary production and respiration. So when we're talking about these processes, we're talking about what makes food webs tick and also what makes the global carbon cycle tick. Okay, and in particular, I'm going to be talking about not just ecosystem metabolism, but metabolic regimes. And what I mean by that is something like this. If you imagine that we've got the day of the year spread out along here from, say, January 1st to December 31st, and we're going to have some kind of picture here of the pattern of these two processes, GPP or ER, and the rate will go up and down through time. Okay, so that pattern of what does GPP and ER look like through time in a particular local ecosystem like the woods outside here, or type of ecosystems like northern hardwood forests, we're going to talk about that as the metabolic regime. We measure that in terrestrial ecosystems these days often like this. So this is a flux tower that uses the eddy covariance method to measure gas exchanges between the vegetation and the land surface and the atmosphere. And by measuring the way that carbon dioxide in particular moves over this vertical space and through time, and by simultaneously measuring things about the way that wind moves through that space, you can do a bunch of math and end up with estimates of, okay, well, what's the net CO2 uptake? That's gross primary production. What's the net CO2 release? That's ecosystem respiration. This is now a pretty standardized thing. People do this in all kinds of terrestrial ecosystems. The towers look really different depending on where you are, because the point is to get them up above the canopy. So in a big mature forest like this, the towers are super tall. You can do this in a marsh where the vegetation's all this tall, and then the tower's only like this tall, right? But the principles at work are basically the same, very standard, and there are huge global networks of people who do this and sort of pool their data together to try to derive insights across the globe. So here's, well, there's no data on it, but this in a minute will be the metabolic regime of a temperate broadleaf deciduous forest like what we have outside. And I'm going to ask you, just as the one annoying participatory thing I'm going to do today, to just take 30 seconds and, like, in your notebook, if you're a diligent student and actually have a notebook here today, or on your palm if you don't, just think about what you think the pattern of just GPP, primary production, looks like through the course of the year outside our room here, right? Just go ahead and draw it on your hand for a second. Yeah. Okay, so here's the answer. The black line is GPP, and we can see how you did on that. Probably a lot of you guessed something that looks kind of like that, right? During the wintertime, there are no leaves on the trees. It's hard to do much primary production at that time of year. The leaves all come on, and gross primary production goes up. There's also a lot more sun here in the middle of the year, and the sun is the fuel for that photosynthesis. And then eventually, towards the end of the year, rates start to tail off because the sun is not up in the sky as much, and the leaves drop, and then there's no more primary production for the rest of the year, right? Ecosystem respiration here in our kinds of ecosystems follows a similar pattern. It's driven in large part by the rate of GPP, although you get significant rates of ecosystem respiration that are sustained throughout the shoulder seasons because those soil microbes and things continue to respire, all the heterotrophs continue to respire, even when there's not much primary production happening. Okay, so that probably feels a little bit familiar if you live in a place like this. You've got sort of an intuitive sense of what the metabolic regime looks like in your home ecosystem. And if it were the case that you lived or spent a bunch of time in a Mediterranean broadleaf evergreen forest, you probably would have a decent sense for this pattern. It's a little bit different, in part because while there's still this influence of the sun being there being more sunlight at some times of year than at other times of year, that's a little less pronounced in some of these ecosystems because they're closer to the equator, right? And also, water availability tends to be a really big and important driver of gross primary production in an ecosystem like this. So you get this kind of spring peak of GPP that happens with spring rains, and then that tails back off as things start to get dry again. And then actually, I don't remember why it peaks again later in the summer. Probably somebody who spent more time in these ecosystems than I have could give us the answer to that. And the pattern of ecosystem respiration is pretty different as well, right? It's a little flatter through the year. So, you know, you get the idea here. You could go to lots of different kinds of terrestrial ecosystems, and people have, and measure these processes. and you get a sense for both the typical pattern in any kind of ecosystem and also the really huge variability there is from one of these kinds of ecosystems to the next in terms of what's the average rate, how seasonal are the rates, how closely are the GPP and ER patterns through the year tracking each other or not, right? Just this tremendous, tremendous variability from one ecosystem to the next. Okay, in aquatic ecosystems, we do something kind of similar. Our flux towers are a little less grand looking. And the process, the kind of mechanics of how we do this is a little bit different. So basically what we're doing here, this is actually Bella Alexa right here on one of the lakes that we work on the Wisconsin-Michigan border. And we have here a little homemade raft that has some wind sensors and light sensor on it, and then hanging from this raft down into the water are sensors that measure water temperature and help us get at the movement, the circulation of water through the water column, and also dissolved oxygen. And so whereas in terrestrial ecosystems, people tend to do this mostly by measuring carbon dioxide, in water, for a variety of reasons, we tend to do this using oxygen. If you remember from intro bio, the basic equation for photosynthesis, you got carbon dioxide on one side and oxygen on the other, right? So you can measure either one of them to get at the rate of that process. We measure dissolved oxygen, and by tracking the change in dissolved oxygen through the course of the day, we can do that same thing. We can estimate what's the rate of gross primary production or ecosystem respiration. So there's been some really nice work on using that kind of approach in stream ecosystems to try to understand what the metabolic regimes of streams are. And this is work by Emily Bernhardt and others over the past, 5 or 8 years or something like that. So here's the typical metabolic regime of a closed canopy stream. This is from a synthesis of data from a couple hundred streams in the continental US, I think. And you can see this is more or less the same scale that I was using for those terrestrial ecosystems. So in terms of area, much lower rates of GPP and ecosystem respiration, and a really different pattern through the year. You know, this might be a stream that's right. This could be Wappingers Creek. You know, it's a creek like that. And we see that the pattern is really different through the course of the year than it is in the adjacent terrestrial ecosystem. And that basically has to do with the availability of light, right? In a stream like that where there's a lot of canopy cover, the time of the year when the light is the most available is here early in the spring, right? The days are longer, the sun is higher in the sky, there's a fair bit of light, and the leaves haven't covered over the stream yet. So the time that we get this peak in GPP happens right around here. Peak in ecosystem respiration is around the same time, and then you have pretty low rates for the rest of the year. One of the really neat things that Emily and her co-authors did in this paper is to take those data that they had on metabolic regimes from a couple hundred streams and try to sort it out into some categories. You know, for those terrestrial ecosystems, we were able to say, okay, well, a temperate broadleaf deciduous forest usually has a pattern that looks kind of like this. A Mediterranean, blah, blah, whatever I said, forest has a pattern that looks more like this. And so what Emily and folks were doing was trying to figure out, okay, well, what are those categories for streams? What are the categories that we should put streams into to characterize what their metabolic regimes look like? And what are the sort of drivers of what those categories are? So for instance, they found that some of the key drivers were how predictable is the flow? Is the flow highly predictable? It's pretty steady throughout the year or has sort of a known pattern? Or is it a much flashier stream where you get, a precipitation event tends to create a huge sort of scouring flow of water, and then, the water's gone again the next day. You might think of a desert stream or something like that. And then the other key variable they identified was light availability. Low availability, something like the stream out here that's very covered by trees, or high light availability, an open canopy stream that's maybe bigger, or maybe is in a place with less tree cover. And so they, kind of using this gradient, they identified these kind of categories of different kinds of streams. They came up with nice names for them, like dark and stormy. So here's a group of streams that tends to have low light availability, it's dark, and also low predictability of flow, it's stormy. And they see particular patterns in the stream metabolic regimes in those kinds of streams. And again, they link this to sort of higher level, kind of watershed level, landscape level drivers of these patterns, right? So ranging from things that are sort of inherent properties of the ecosystem, like the size of the river, how much shading it has, how much ice and snow cover there is, and then also moving on to some things that are more human-driven, right? To what extent is there sediment pollution or deforestation of the riparian area, and some potentially climate-driven things like, how are reduced snow and ice cover with climate change going to affect that, okay? So the thing I'm driving at here is basically like, we can take these patterns of GPP and ER in lots of different places, measured in lots of different places, and we can use them to try to understand some kind of general patterns. How do ecosystems differ and how do they not differ, and what are the drivers of that? Okay, here's in contrast what we know for lake metabolic regimes. We can't really do that same thing. You know, Emily put together this really nice framework for thinking about, okay, well, what are the patterns that we see? What are the drivers of those? In lakes, we can't do that, at least not yet, I would say. And that, when I realized that, was a little surprising to me, in part because the advent of using these methods to measure GPP and ER probably got going a little faster in lakes than it did in streams. The lake folks really kind of led some of this work, and the stream folks jumped in a little bit later. And somehow the lake folks, and I'm kind of the first culprit here, were just a little slow and dumb about really thinking about these higher level questions. So we have pulled together some data like this. This is data from a paper that I and many others, including some folks in this room, wrote a long, long time ago, pulling together data from 25 lakes around the world. And in each one of these panels, each panel is a lake, and the data, the points up on top are gross primary production, and the points down in the bottom are ecosystem respiration. And you can see that there are a bunch of different patterns here in terms of that metabolic regime through time. When we wrote this paper, that wasn't a thing we were focused on at all. We really were much more focused on, well, what are the drivers of the day-to-day variability in these rates? And if we kind of take average rates over the course of a whole year, what drives the difference from one lake to the next in those average rates? And we weren't really thinking at all about, okay, well, what is the actual pattern through time here, and why does that differ? And I should say, this is a project that came together through the GLION network that Kathy and Paul have been really active for so long in helping to kind of nurture this grassroots network of folks who produce these kinds of data and ask these kinds of questions. You all probably have heard a lot about GLION. And in this project, we kind of tapped into that GLION network to pull together the people to think about these ideas and the data to let us really test those ideas. So this is maybe a first kind of possible crack at being able to think about some of those questions that I talked about, although we never did it. More recently, Bella, when she was here, sort of tapped back into the GLION network to pull together a similar but even larger set of data. This is data from just under 60 lakes around the world, and the map there shows you where those lakes are located. Again, to ask a different set of questions. Bella and I and others who were co-authors on this paper weren't really focused on those metabolic regimes, but instead, in this case, on testing a new kind of theoretical mathematical model for drivers of lake primary productivity. This, I think, is the largest agglomeration of lake metabolism data that exists anywhere right now. It's a huge set of data. It was a huge undertaking for Bella and all her co-authors to pull this together and quality control it and like kind of make it ready for analysis. And it's a great resource. And yet it's only 58 lakes. And so that was, again, as we were thinking about putting together this proposal, that was part of what we were thinking about is, okay, well, could we go bigger with this? And what would it do for us if we could go bigger? Okay, so as I said, Bella didn't think much about these kind of metabolic regimes, but we generated all the data. The underlying data that go into her analyses are these daily rates of GPP and ecosystem respiration. And so as we started to think about the ideas for this proposal, one of the things that we did is actually go back into her data and make these plots and look carefully at, okay, well, what are the patterns that we see in these different lakes through time? So what you see here, again, the black lines are GPP, the cyan lines are ecosystem respiration. And each one of these panels here is a latitude bin. So this is between 35 and 45 degrees south latitude. Here's 35 to 45 degrees north latitude. You get the idea. And then in each panel, there's one line for GPP in a given lake, and somewhere down here, a line for the ecosystem respiration in that given lake. So what I'm going to do to make this a little bit more manageable is just zoom in on the lakes here in this latitude band. This is more or less where we are, a little bit north of where we are maybe. What's our latitude? Anybody know offhand? Oh, that's shameful that none of us know what latitude we're at. Okay, we'll work on that for next time. I don't know either. Okay, so we're going to zoom in on that. And if we take each one of those lakes that was in that panel and we just look at each one together, here's what we see. That's a little overwhelming, honestly. There's a lot of squiggly lines on there, so I'm going to focus your attention on a couple particular patterns. So this is Cranberry Lake or Cranberry Bog and Northgate Bog. These are two small lakes that I work on in northern Michigan. They're a few kilometers away from each other. Like you could get from one to the other in a car in a minute or two. And yet they have really wildly different patterns of gross primary production throughout the year, right? Like if you look at sort of the seasonal trend and where this black line goes, it looks really nothing at all like the seasonal trend in that black line, right? Why is that? Those lakes are right next to each other. Why do they have such different patterns in gross primary production? We can do a similar thing. Here's Cranberry Bog again, and Crampton Lake, which is, again, within a few kilometers of Cranberry. It's a little bit bigger. In this case, you can see that the pattern GPP through the year is a little bit more similar. They both seem to have this peak that occurs here, you know, kind of early summer, and then a decline after that, and then sort of this maybe early fall peak that happens. So a lot more similarity in the pattern of GPP. And yet the ecosystem respiration trends in those two lakes are wildly different, right? Here in Crampton, as you go through the year, basically the ecosystem respiration is always increasing. It's moving away from zero here through time. Whereas in Cranberry, you've got something more like a mirror image of the pattern of gross primary production, right? Does that make sense? I've looked at these a lot, so this just feels kind of natural to me, but maybe it's a little slower for you folks. Okay, so like one of the things that really strikes you when you spend a lot of time staring at these plots, as I have now done, is like the longer you look at it, the more puzzles start to emerge from it, right? You can look and think, oh, these lakes are right next to each other. Why do they look so different? These lakes are super far apart, and they don't seem to me like they're that similar in the ways that I usually think of. Why do they have such similar patterns, right? And so what we've sort of been wrestling with is, okay, how do we make sense of this diversity of patterns that we see in ecosystem metabolism? Here's kind of where we have ended up with that, or at least for now, where we've ended up with that. I'm going to walk through for a minute or two here sort of the conceptual framework that we're working with now and that we're hoping to be able to test should we get some money for this proposal that we put in. And the starting point for that is that, okay, well, we have some pattern of GPP through the year, and that's at least one of the influences on ecosystem respiration, right? Gross primary production does two things. Number one, it means there's cellular activity of the primary producers. That means that those primary producers have to respire, so there's some inherent link between those two processes there. Also, primary production creates organic matter, which then heterotrophs can break down. So again, there's some inherent link between those two processes. And if we think about sort of the most proximate drivers of those processes of gross primary production and ecosystem respiration, our argument is that basically there's a very common set of proximate drivers that you can think of whether you're in a lake or a stream or a forest or a grassland or whatever else, right? There are things like, well, what are the energy sources that are available? You know, either light for GPP or fixed organic matter for ecosystem respiration. There are some key reactants that go into these processes, like what's the availability of CO2 or water. There are some things that regulate the rates of primary production or respiration at kind of a cellular level, like key nutrients that are cofactors in the pigments that actually capture photons from the sun and capture that energy, right? Or temperature, which we know has a big control on the rates of lots of cellular processes. And then there are losses. And here's where maybe one difference is between terrestrial and aquatic ecosystems. In both terrestrial and aquatic ecosystems, we have to worry about losses of the primary producers or the heterotrophs, right? That when we have fewer of those organisms doing the process, the rate of the process should change. In aquatic ecosystems, we worry about another kind of form of loss of the primary producers, which is flushing. Water moving through the system can wash those primary producers out faster than they can replace themselves. That doesn't happen so much in a forest. Okay, so those are the proximate drivers. We can also kind of think backwards to, okay, well, what are the things that ultimately create the patterns that we see in some of these proximate drivers? And for lakes, we think about those as things like, what's the lake morphometry? How deep is it? What's the area? What's the watershed like? Is it a big watershed or a small watershed? What's the land use on that? Is it forested? Is it agricultural? Is it urban? All these things are linked to the availability or the kind of patterns in these proximate drivers, right? Things like latitude ought to be important because the latitude plays a big role in how much light you have at what time of year. And then in between these kind of ultimate drivers and proximate drivers, we think that there's a set of kind of common key intermediate drivers which are driven by these ultimate things but have a big effect on the proximate drivers. And in lakes or aquatic systems, those are patterns of stratification or mixing of the water and patterns of residence time as well. And I'll talk a little bit in more detail about what those things are and what they mean. To do that, I'm going to pull out one particular kind of set of connections through this conceptual model, which is to think about how a variety of things, including lake morphometry, watershed characteristics, and latitude, via, among other things, their effects on stratification and mixing, might influence energy sources, in particular light, that end up controlling GPP and ER. So I'm going to walk a little bit slowly through just kind of one set of possible connections between these ultimate drivers and those proximate drivers and the patterns we think we should see. Okay. So here's a start of that, and to think about the availability of energy, the input of light energy to a lake ecosystem, a good place to start is to think about, okay, well, the sun hits the surface of the lake, and there are patterns through space and time and what that looks like. I've talked about this a bunch already. Here's some data. This is from Began and Townsend is like an intro ecology book. I used an older edition of this textbook when I took intro ecology 1,000 years ago, and this is the updated version of it. So these are like literally textbook patterns in ecology, right? Each one of these figures here, this is for Bergen, Norway at 60 degrees north, and this curve here is showing you for an average day in the month of May, if I drew a vertical line there, what's the availability of sunlight, of solar radiation through the course of the day, right? So your average day in May, you get a bunch of sunlight. Your average day in January, you don't get very much sunlight. Does that make sense the way I'm explaining that? Yeah, okay. All right, and unsurprisingly, if you go to Portugal at 40 degrees north, you see higher curves there. If you go to Pune, India at 18 degrees north, you see higher curves there, and you see way less seasonality, right? So this is not surprising. This is the pattern of solar radiation across the globe. It's one of the major, it's maybe the major driver of patterns of metabolic regimes in terrestrial ecosystems. For aquatic ecosystems, that's sort of just the tip of the iceberg, right? Because that light hits the surface of the water, but water is a very, very strong absorber of light energy. And so as you move down through the water column, what matters is not so much, it matters some, but not so much how much light hits the surface, but how that light is extinguished with depth. And we know that the rate at which that light is extinguished with depth varies quite a lot from lake to lake, even from between lakes that are right next to each other. So here are data from a couple of those lakes that I work on a lot in northern Michigan. One of them has pretty dark colored water, one of them has pretty clear water. And what we're seeing here is as you go deeper into the lake from 0 meters, the surface, down to 5 meters or more, what percent of the surface, of the light hitting the surface is still available. And you can see that in the dark lake here, by the time you get down to, what, like 1.25 meters or something like that, you're at something like 1% of the surface light is left available. So, you know, that's like this far down into the lake, right, from the floor to about here or something like that. You're down at 1% light by the time you hit the floor there. 1% light is our usual measure of kind of the minimum amount of light that's required for photosynthesizers to make a living, for the benefits of photosynthesis to outweigh the costs, okay? And so the available water column for light in a lake like that is very shallow. In this clear lake that, again, is 1 kilometer or 2 away, you can see that you can go down to 5 meters or actually even deeper, and you still have enough light left for photosynthesis to be happening, right? So huge differences from one lake to the next in how rapidly that light is attenuated with depth. Okay, so there are differences from one lake to the next. We also know that those differences from one lake to the next in how dark or clear they are pretty predictable. They're at least some predictable, right? So here is a measurement of dissolved organic carbon concentration. That's one of the major drivers, not the only, but one of the major drivers of how clear the water is, how rapidly the light is extinguished. And this is a synthesis by Maggie Xenopoulos showing that lakes with a big watershed relative to the size of the lake tend to have higher concentrations of DOC, right? That makes sense. That DOC comes largely from the dissolution of terrestrial organic matter in rainwater in the watershed. And so if you have a big watershed feeding into a small lake, you've got a whole lot of organic matter out there that can be dissolved and end up in the lake. Okay, so again, we're trying to work from what are these proximate drivers on these processes? Can we think about the sort of higher level, at the landscape scale, what are the things that really ought to create differences from one lake to the next in these patterns? Okay, it gets a little more complicated than that because not only is the light extinguished with depth, but also the surface waters of lakes are mixed continually by wind energy. So if you're a photosynthesizer that lives in a lake, you're a single-cell alga or something like that, it doesn't matter so much what the light level is here, where you are right now at this moment. What matters is that over the next 5 minutes or hour or a few hours, you're going to end up circulated over this whole range of depths, right? And so the amount of light that you see on average is the average of the light at all of those depths, okay? Here's a little glimpse into what that looks like. The colors, so this is data for whatever this is, a week or 10 days or something in the fall. Again, in one of these lakes that I do a lot of work on. We've got the surface of the lake here. We're going down to 5 meters depth again. And the colors are indicating the light availability. And so what we've done is to take, okay, what's the rate of light extinction? And also how deeply is that lake mixed? And so then we can calculate, what's the average light that a algal cell sees over this interval of 10 or 15 minutes or whatever. And you can see a couple of patterns here. One is that during the daytime, you have brighter colors. During the nighttime, you have darker colors. That's not really a surprise. There's more light during the daytime than the night. You can see some day-to-day variability. So over here, these were a couple of really cloudy days. There's not much light hitting the surface, and so there's not much that gets down into the water column. But other than that, this lake is fairly stably, over this time period, mixed down to about, I don't know, 3¼ meters or something like that. So there's not much variation day to day in kind of the amount of water column that's available to those photosynthesizers. Did that make sense? Okay. All right. So let's contrast that with, again, a lake that's quite nearby. And I'm going to say, just by way of explanation, the gray areas here are time and space periods where the amount of light is so low that it kind of falls off of our color scale. So it just shows up gray here. You can see some similar patterns, right? Like you see the daytime and the nighttime. You see that these couple cloudy days over here, there's not much light availability. One really notable difference is that this lake, because it's small and wind protected, and because it has fairly dark water, and so it absorbs heat energy from the sun very quickly, tends to microstratify each day. The surface water heats up so much that you get a density stratification between, it sets up at maybe, I don't know, 1.5 meters down, and by late afternoon, it's strengthened to the point that the water's only mixing down to whatever that is, 3/4 of a meter deep, something like that. So every sunny day, the stratification moves up towards the surface, and then at night, as the air cools down, the water then cools down, and that density stratification breaks down, and the water mixes back down again, okay? So these two lakes, again, right next to each other, huge, huge differences in the average light climate that an algal cell experiences in one lake versus the next. Okay, and we know again that these differences in mixing regimes they're predictable more or less from lots of kind of higher level characteristics of these ecosystems. So this is a famous figure from a limnology textbook showing how the latitude that you're at and the altitude influence in a sort of a coarse way the general type of mixing regime that you have. Some new work by Meredith Holgerson, our neighbor at Cornell, has sort of extended this idea to largely to smaller lakes, to ponds, and thinking about how lake area and depth control the mixing regimes that we see. And so again, there are these sort of predictable patterns. You could say to me, okay, well, here's a small lake at 30 degrees latitude and near sea level. What's the mixing regime probably? And a limnologist, after a little bit of thought, would be able to give you the answer to that, or for any other kind of lake around the world. Okay, so that was a long-winded way to drive at what we're thinking of as kind of the conceptual model here, which is that we've got regional kinds of drivers, things like latitude, right? If I'm at a lake here or a lake a kilometer away, they're at essentially the same latitude, it doesn't matter. And so something like the amount of light that hits the surface of the lake is going to be identical for those. So you might imagine that, okay, well, here's a tropical lake, there's not a whole lot of variability in the incoming solar radiation through time, and so the GPP does this. Here's a lake kind of more in the temperate zone, here's an Arctic lake or something like that, right? So there are these regional drivers that might create these kind of coarse differences in the metabolic regimes through time. But then there are also going to be these local drivers that might differ a whole ton, from one lake to the next within a couple kilometers of each other. And so those might sort of add some other dimension of variability into these patterns, right? I don't know what local driver I was thinking of here, but it's one that creates wiggle for some reason in that pattern, right? And, oops. And so if you kind of play that out, you end up with, again, just for a couple drivers, you end up with all these different patterns. If we think about the fact that there are multiple regional drivers and multiple local drivers, you can imagine how you might end up with this kind of spaghetti mess that resembles what we saw when we looked at those data that Bella produced, right, from those 60 lakes around the world. And so the idea is that, okay, well, if you could look at the spaghetti mess and then kind of think backwards towards what the drivers are, could you try to start to make sense of, okay, well, what are the important drivers, and can we start to classify what the different kinds of lakes are? Okay, and so that's one of our goals in this project, is to take the spaghetti mess and say, okay, well, type A, here's a kind of lake, here's a kind of metabolic regime that we see, and it happens in lakes that are small, deep, and located in the tropics, or it happens in small, deep lakes no matter where they are in the world, right? Things like that. Okay, so for the last bit of the talk here, the thing that I want to do is tell you a little bit about some of the hypotheses that we're thinking about testing that kind of come out of this conceptual model. And I'll show maybe just a little bit of data to kind of tease where we might be going with some of those hypotheses. Here's one of them, and I kind of hinted at this a little bit in what I just said. The hypothesis is basically that lake biomes, if you think of things like temperate deciduous forest, right, or Mediterranean grassland or whatever as biomes, the biomes of lakes ought to be weakly clustered in space. That's our prediction. This is a map of terrestrial biomes from NASA, and these are things that are going to be familiar to you. Like, I think this is rainforest in here, unsurprisingly, in the Amazon. Here's, I don't know, temperate grassland or something like that. You get the idea. These things are, they occur in big blobs, right? Like the tropical rainforest here is in this big blob. The grassland's in this big blob. Our hypothesis is that if you were to do the same thing in lake ecosystems, they don't blob like that, right? So if you imagine you've got type A of lake in black and type B of lake in green and type C in red, it might be that here in whatever this is, the Canadian prairies just shy of the Rockies, that you see black lakes and green lakes and red lakes right next to each other. And you see the same thing down in, I don't know, Indiana or something like that. And to some extent, you see the same thing if you're in Southern Africa, right? Like, while there may be some kind of types of lake metabolic regime that are regionally specific, it may be that because of the influence of these local drivers, which can vary a lot, between two lakes, whether they're here in North America or in Africa, that we see sort of similar typologies as we go across the globe, and we don't see these big blobs of like, okay, well, every lake that's located in northeastern North America is the same lake biome. Does that make sense? Yeah. Okay. So that's one of our predictions. Another prediction is that there are differences in the day-to-day variability of these metabolic rates, and that those ought to be driven by things like hydrologic and thermal stability. So here's an example of what I'm talking about. This is Golensee, which is in Denmark, I think, and Lake Sunapee, which is a lake that Kathy has worked on a lot in New Hampshire. You can see that the average pattern of gross primary production in these two lakes through the year is quite similar. They peak at about the same time. They're at about the same magnitude. But the daily variability around those kind of average seasonal rates is hugely different, right? There's a ton of variability here in Golensee, and there's much less variability in Sunapee. Why is that? To get some insight into that, I'm going to walk you through some results from Jake's work from a number of years ago now. And what we're looking at here for starters is the input of organic carbon, dissolved organic carbon from the terrestrial landscape over the course of a couple years here. And this is for a particular lake that has a hydrologic residence time of 36 days. Okay, so that means on average, water molecule spends about 36 days in the lake. Each one of these dashed lines is a precipitation event. And so you can see that every time there's a precipitation event, there's a huge spike in the input of organic carbon from the watershed into the lake. If you go to a couple lakes that are right nearby, but that have very different residence times, so here's East Long Lake, it has a residence time of 300 days, and Crampton Lake, which we talked about earlier, which has a residence time of over 720 days, a couple years. Those same precipitation events produce much different patterns in those inputs of organic matter, that stuff that's the fuel for respiration, right? And that basically happens because there's so much more volume of lake to absorb that input here in Crampton Lake relative to the watershed that's delivering that stuff, that you don't really see the spike in the input there. There's just not that much new input of organic matter relative to what's going on in the lake. Okay, and then the other thing that Jake did was to take these same lakes and, again, these same precipitation events and look at the patterns in this black line up here is GPP. This black line down here is ecosystem respiration. And for now, we're going to ignore the gray line, but that's NEP, net ecosystem production. And you can see that right after one of these precipitation events that delivers organic matter into the lake, you tend to see a spike in the rate of ecosystem respiration. And Jake actually did a nice thing where he sort of calculated the kind of underlying trend in ecosystem respiration and then was able to isolate, okay, well, what's the effect of actually that fresh input of organic matter that happens as a result of the precipitation event. I'm not going to talk about that. I guess I will in a slide or two. So he did that, trust me. Here's the pattern for the other two lakes, and this probably isn't going to surprise you. Just as we saw that those precipitation events cause really different spikes in the inputs of organic matter, we see that they have really different impacts on the spikes in rates of ecosystem metabolism, right? Down here in Crampton Lake, it's almost impossible to detect any signal of those precipitation events on the pattern of ecosystem respiration. Okay, here's a synthesis of that. This is what's the effect on, let's talk about this as ecosystem respiration per event for these three different lakes. One with a really short residence time, sorry, this is residence time here. one with an intermediate residence time, one with a long residence time. And you can see that the average effect of those events on the pattern of ecosystem metabolism declines as you go to these really long residence time lakes, okay? You can think about kind of a similar thing with that example I talked about of the lake that's got really dynamic surface stratification and mixing, or the one that has much less dynamic surface stratification and mixing. These things that are sort of controlling the, I don't know, the size of the reactor in which these biological processes are happening, right? Or like how fast we move the water through that reactor. Those ought to have big impacts on the rates of ecosystem metabolism. Okay, here's a first look at whether we actually see a pattern like that from the data that we're starting to assemble for this project. This is, again, for the 60 lakes that Bella pulled data together for. And this is the variability in GPP from day to day against residence time. And you can see something like that pattern, right? We tend to see higher variability day to day at really short residence times and less variability day to day at really long residence times. Okay, here's another one that Tara was really involved in helping us think through. This is thinking about the clear water phase in lakes. So depending on where you are in the globe, it's a pretty typical pattern for lakes in the springtime to have a spring bloom, so they get greener. The biomass of photosynthesizers goes up. And then often that spring bloom is followed by what's called the clear water phase. The water really suddenly and really noticeably gets a lot clearer because the population of algae has kind of crashed. One of the main drivers of that crash, usually we think, is the most important grazers in the ecosystem, things like Daphnia in particular, which are filter feeders that really control the populations of algae. And so the idea here basically is that we can think about the magnitude of this decline, whether this kind of spring clear water phase in GPP from this spring bloom down to the clear water phase, whether that occurs and how big that clear water phase is, how big the improvement in water clarity is, ought to be a function of, again, a really local driver, which is what does the food web look like? Are there lots of Daphnia in that food web or not? And so the idea here is that, okay, if we go from lake to lake and we quantify the biomass of Daphnia, which is, again, usually the most important grazer, that the size of that decline, that red arrow in that last figure, ought to be related somehow in one of these patterns to the biomass of Daphnia. Okay, and then I think I'll do just one more of these hypotheses, and then I'll wrap up for you. And this one's sort of related to climate change. There's a nice study by Catherine O'Reilly a number of years ago now, and there's been other work on this since showing changes in the surface water temperatures of lakes through time. So any point that's in red or orange here is a lake where the surface water temperature has been, the trend has been increasing. The blue ones are decreasing. You can see that in general, lake surface waters are increasing in temperature. And in fact, the rate at which that's happening almost always exceeds the rate at which air temperatures in the same place are increasing. So lakes are responding in terms of their temperature very quickly to climate change. That matters for ecosystem metabolism because rates of ecosystem respiration in particular, so the blue stuff here, and to a lesser extent, gross primary production are temperature dependent. And because the rate of ecosystem respiration is more strongly temperature dependent than gross primary production, our prediction is that if you were to be able to follow a lake through enough time to see a substantial change in the average temperature, which we can probably do with a lot of these data sets now, and or if you amass lakes where you have enough variability in average temperature, which is kind of what we've done here, that we should see stronger increases in ecosystem respiration than increases in GPP as a function of temperature. And that means that these lakes should be becoming, long story short, bigger sources of carbon dioxide to the atmosphere in general as the temperature warms, okay? That's a simple prediction that's built largely on like metabolic theory of ecology, if you know about that. There are some reasons to think that that might not actually work. There's some kind of potential complicating factors. This little bit of data that we've accumulated so far suggests it probably does happen, but we're interested in expanding this data set and seeing whether that's true or not. Okay, so that was sort of a blast through what we've been thinking about with this set of ideas and some of the particular hypotheses that we want to be able to test. How are we actually going to do that? The idea, one of the core parts of this proposal is to put together a thing that we're calling the Global Lake Metabolism Repository. It's got a great acronym, GLAMR. If you say it right, yeah, that's good, right? And it feels like it ought to have a really good logo. This is like 3 minutes in PowerPoint worth of logo, but it feels, it's glamorous, right? We can get a promo from a movie star or something. It's a good acronym. So the idea is to take all of these metabolism data sets that are generally individual investigator-led. So somebody working on some lake somewhere maintains the infrastructure to be able to estimate these kinds of things. And there's like hundreds of those people around the world. And despite the fact that Gleon is this great talking shop for those people to get together and work together, there is not a sort of central repository for anyone to go and access those data, right? In contrast to what we have with terrestrial ecosystems, things like Ameriflux or FluxNet, there have been for many, many years now sort of repositories for this data that make it possible for anyone to kind of do synthesis analyses across a really broad scale. So our goal is to try to basically create the same thing for lake ecosystems. This is a little snapshot of some of the data that we know are available and could potentially be brought into a repository like this. A little bit of quick searching and math suggests there's probably hundreds, 200, 600, somewhere in that neck of the woods, separate lakes around the world where data like this exist. And on average, probably at least 5 to 10 years of data for each of those. So there's potentially just this vast wealth of data available to start to do these kinds of syntheses. And that's what we're hoping to do as the first part of this project, and then building from there to kind of answer some of these ecology questions. Okay. I think I kind of said this. Like, the idea is take all the work that's being done by individual investigators in a gazillion places around the world, try to bring it together so we can do some of the synthesis and do some really great science. I'll say thanks, and I'm happy to take any questions.
Just a reminder, Chris, to repeat the question after. Yeah, you got it.
Yeah, thank you. Vanessa.
It was a great talk.
Thanks.
And I was wondering if is Daphnia the principal grazer in all these lakes across the globe? If not, what are you thinking it will be important to measure in those lakes?
Yeah. Sorry, the question, I almost forgot right at the beginning. The question was about whether Daphnia is the principal grazer in all these lakes around the globe, and if not, what else might we measure? I'm going to answer, and if I get this super wrong, Tara's going to jump up and shout or something. By and large, Daphnia seems to be the only one that really is an efficient enough grazer to really produce these kind of clear water patterns where it really knocks down the population of algae. Tara's nodding.
Large-bodied Daphnia.
Large-bodied Daphnia. There are a variety of species of Daphnia, but big ones in particular. So our idea is that as kind of a first cut, you could look maybe even just at the presence or absence of large-bodied Daphnia, and that might be a sufficient indicator, or maybe biomass of Daphnia. I think other zooplankton are also important grazers on algae, and there are places where like mussels, filter feeders can be important controls on algal biomass. And so that's not going to explain all the variability. And our hope is that, a big idea with this project is kind of an if you build it, they will come thing. Like, if we start to assemble the data to make these kinds of analyses possible, that people will start bringing to the table better zooplankton data sets and things like that so that we can ask more nuanced questions. But we think it ought to be pretty easy to get at least, are there large-bodied Daphnia there or not for a whole bunch of lakes? And so that lets us ask kind of the first-order question. That's the goal. Does that answer your question? Yeah. Jazz.
Yeah, thanks. That was really fun. I'm wondering, what sort of methods do you currently have planned to convert squiggles into lake
Yeah. The question is, how do you convert squiggles into lake types? There's a couple things. One of the main things, or the one that sticks in my mind the most, is a set of tools called time series clustering. And I haven't actually done any of that yet. I've just read some papers that have done it. But my recollection is that there are some like sort of likelihood-based approaches to that, and then also some machine learning type approaches to that. And they're like some of the stream metabolic regime work actually has used those techniques. And they seem to work okay to identify clusters. Do you have other ideas?
Well, I wondered if it would be a fun use of like image-based machine learning. You can just feed an algorithm.
Oh, that's interesting.
Ask it to classify it into groups.
Yeah, so I'll repeat that for the online audience. Jazz's idea there was, could you just use image-based kind of machine learning to sort out what are the flavors of squiggle that you have available? That's a neat idea. I hadn't thought about that at all. Yeah, thanks.
You mentioned a lot of the proximate and underlying drivers, and you had a number of the underlying drivers that you said could all be important on how it affects things. The plots that you showed us seem to say that dissolved organic carbon has a much higher importance at predicting GPP and respiration than the others. Have you run a PCA array on forest yet to see if that actually holds true?
So the question is, there's an observation first which says that it seems like DOC is an important driver of a lot of these processes. And the question is, have we done any analyses like a PCA or something to see if that kind of jumps out as a major driver? I'm going to answer that question in a couple ways. One is that DOC is near and dear to my heart. And so partly you're seeing evidence of the fact that it's quick and easy for me to think of examples where DOC is the driver. A slightly more scientific answer than that is we do think that DOC is an important driver of GPP in particular because it has big influences on the light climate and on the heat environment of the lake, and therefore stratification and mixing. And actually that model that Bella was testing that I mentioned in that paper is a model in which DOC is a major driver of lake gross primary production. But we haven't done the analysis you mentioned yet. It's a good idea. I'm not sure really that DOC would emerge as the only, like latitude has got to be an important one, right? Like the latitude drives how much light hits the surface, and it has a big effect on the mixing regime itself. So like latitude has got to be important, right? Watershed characteristics have got to be important because they determine what's the nutrient budget of the lake, right? Are there nutrients there to support primary production or not? So I'm not convinced that actually when this is all said and done, I think DOC will be one of the important drivers. I don't think it'll be the important driver. Ask me in a few years. Jane.
This is really fun to think about. I'm curious, you kind of alluded to this, but I'm just kind of curious how often the GPP-ER ratios, like how similar they are between terrestrial and aquatic systems, or are they just like always acting independently, and maybe what scale? Because you show like a temperate broadleaf, but that's a pretty big classification, and so yeah, like can we ever say like how closely those two are linked in tracking each other?
Yeah, I got started thinking about answering your question, and so I'm not going to do a great job repeating the question. But basically, it's like how closely are the GPP and ER relationship in a terrestrial ecosystem linked or not to what you see in the aquatic ecosystem that is embedded within that context. Is that okay? I don't know. That's an interesting question. There's a figure in Emily's paper that I was citing there that they pulled some Ameriflux data, I think, and show kind of the distribution of rates of GPP and ER for terrestrial ecosystems, maybe across the US, and then the distribution for stream ecosystems. But off the top of my head, I don't remember that they did that kind of one-to-one matching. And I haven't thought about that question explicitly, and it would take me more time than I'm going to take right now to come up with an answer, but it's a good question. Let me think about it some more. Remind me. Can I do one more from here?
Sure.
Mark, do you still have a question?
That was more or less my question.
Okay, good.
So we have a question from the chat. Taylor says, "Hi Chris, great talk. I'm wondering if you know how well CO2 emissions estimates from lakes correlate with metabolism fluxes calculated from oxygen measurements, and if there are certain types of lakes where these trends seem stronger than others."
Paul, Kathy, can you think of anybody who's done that?
There are some flux towers near lakes.
Yeah.
Concord has one, and I would look there first.
Yeah.
Wouldn't it be an indication of respiration coming from?
Yeah, so that's a good idea, and I'll repeat it for Taylor since she can't hear you. Kathy points out that there are at least some places where folks have deployed flux towers kind of next to lakes to get the terrestrial environment, but also the lake, and so you could look at places like that. An example is coming to my mind, which is a study from an experiment. Daniel Schindler is the lead author on this, where they looked at kind of the carbon emission implications of a food web change that changed DPP and ER in a lake. So that's a link. I don't know if there's a general, I don't know if anybody's tried to do that generally. It's a good idea, Taylor.
Great. Well, with that, let's thank Chris again for a great talk.